All right, so we got a little bit of planning to do. So next week, all right, next week is gonna be a pretty busy week, right? So your short essays are gonna be due on Wednesday. So for that one, if you go on Google Brightspace, you can click on the calendar and it'll tell you exactly that prompt, that first question asking about emerging property. For that, be creative in your writing. I don't want to hear about the car analogy or the bread analogy. Otherwise, I read the same thing like 40 times and I'm tired of it. So make them something interesting, right? And remember, the whole thing about emergent properties is like, you can look at all these little things, right? And you can say them by themselves. It's kind of like there isn't one of them that's responsible for that effect that's possible, right? Only when the stuff comes together is it that it's like, that's what makes it, gives it that extra skill or something that comes out of it. Like, a bad one could be like, kind of like, if you imagine like going to a party, right? It's like there's not that one person who's like, hey, that person in the room by themselves, it's gonna be a good party because they're there by themselves. Only when all the people come together, then it's like, hey, now we have a friend, and you have a good time, right? So that good time isn't inherent to somebody. It's only when you have that group behavior that you can make that happen. Does that make sense? So when you guys bring those things up about your emergent properties, I don't care what it's about, just make sure you make that big distinction, right? Which is like, hey, this property that if you try looking at individual units, to be honest, you're not gonna find it and you can't figure it out. Only when you look at it as a group, before you actually get possible. Does that make sense? Yeah? Not?
Do you want that like single spaced or double spaced?
I'm cool with double space, that's fine. The only thing I don't like is when you guys are doing your like on the left, most people like they put their name, they put the class, they'll put my name, they'll put the date, and then they'll make all of those double spaced, and then it ends up being just half a page of introduction, right? So I'd rather you fit all those things, put them single space at the top, so compact that, and then it'll be like about 1 3/4 of a page right. If you want to leave it like that and you leave that however much space you take up on top, give me that on the third page. Yeah? More questions? My review, I have some reviews coming up, so I'm not sure when, but you guys might have, we might have guests, other professors are gonna come here and kind of look at my lecture. When they come, just act normal, just do everything like you do in our regular class. To be honest, I'm hoping they get here, they just hide in the back, so it's normal, just do our regular stuff. With that being said, for me, it's like I'm still working on, getting my slides right there together, so it's like, sometimes I think I did to you last week, a little like a comment card. Like you guys give me like, what's it called, you can give me comments about the class and you won't have your name on it. It's just like, hey, what are things that are really helping you out? I'll play around with how I structure the class, with kind of the way I present things. And then it's like, hey, tell me the things that you enjoyed that really helped you versus the things that like distracted you or weren't really that helpful. Yeah. So I'll be doing a few more of those. What I'll do is I might put 1 or 2 this week or next week. Probably I'll do it after the exam. It'll be a good time to do it. And what I do is you guys will do the little questionnaire, trying to give me some clues, things that'll help you. And then on the other side of that, I'm just gonna get a sheet like this. I'll write everyone's name, and that way you guys get credit for being here, but then the comments are kind of like, those are separate. Like I don't want this to mix. Does that make sense? Yeah. All right. Cool. So that being said, also forgot you guys have your midterm is gonna be on Friday, right? So on this Thursday, on that Wednesday, is normally when we do a review in the class. My thing is, I almost want you guys to study. Like if you're thinking you're about to take the test on that Wednesday, right? I need you to go and then just in your head, prep yourself to be like, Wednesday's my midterm, Wednesday's my midterm, you just start studying for it. That way when you come to that class, if there's stuff that when you're studying doesn't make sense, or you're like, hey, I really can't narrow down the important things of a big topic, that's what I want you guys to bring to me on that class. I'm gonna put up slides where, again, I'm gonna go through all the slides that we've gone through so far, and I'm gonna trim a lot of them. Like I'm only gonna leave like the main points, right? I sometimes when I'm looking at it, I'm reading slides off my new like deck of slides. I'll get rid of stuff where I'm like, hey, this is all being presented somewhere else, and I'll cut up huge chunks. Doesn't mean they're important, I just know that they're in the background, right? So if you're reading through that shorter list and stuff doesn't make sense, you can always go back to the bigger PowerPoint and kind of go up, look at the slides around it, and get a little bit more details. And then I'll also put up an extra PowerPoint where it'll be open and you guys can edit it. So it's like, if there's any questions you have, or if there's something that when you're studying this slide, you guys are allowed to just throw them in there. And when you come to class that day, obviously open up that PowerPoint and you can go through whatever questions that posted. If they're easy for me to answer right then and there, I'll write them in the answers in the PowerPoint. If not, we'll talk them here in class. Yeah? So they're working pretty good? All right, cool. So give me those lessons by Wednesday. I think it's Wednesday night. So, you know, I'll do that with me, but I prefer you guys doing this day, like if you're ready for the midterm. All right, today we're talking about the endomembrane system. So we're gonna look at this video twice today, one at the beginning and one at the end. It's gonna sound pretty familiar because things I've been saying, I said everything on how those little, basically those little fat bubbles work inside of our cells, right? Other little chambers and then we can move things across. Here we're gonna track a molecule, kind of getting us our journey from the ER and getting us out to the Golgi and actually getting us outside of the cell, right? So we'll take a look. We'll listen to the first time, next time I'll turn up the volume, but we'll check it out.
Protein processing and
I'll have to cut it a few times. All right, so here, just so you guys look like this, so we're hanging out, basically, this is the edge of the ER. And the thing she doesn't tell us that well is this red thing back here, that's our mRNA, right? So that's the sequence that gets translated, and it's gonna make these little purple things, and that's your protein. Let's go back, sorry.
protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are two types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down.
All right. You can see she started a little bit halfway through what I normally like to talk about. For me, when you guys are thinking about the journey, we always start with the nucleus, right? So we're saying nucleus. And here, remember, the big thing we're imagining here is that this is the DNA cell. From that, the message that we send out is basically our RNA. And this is the one that's going to leave the nucleus and actually makes it to the ER. So when we look at the ER, right, this will be our second stop. You can kind of see that we have this thing built around, or like it's structured around your actual nucleus, right? When you're looking at these little lines, I want you guys to know, remember that these blue lines basically represent a phospholipid bilayer. So even though there it looks all fancy and super complicated, like in my head, I'm thinking almost like it's just a little bit like a little fat bubble, right? That's the way I think about it. The ribosome is going to come sit like this. The RNA basically swings through this part, and we're making the protein inside of this structure. Right? Okay, I'm gonna my colors. So we're basically putting our protein inside of the BIOL itself, right? So then at the next level is we're going to jump over to that Golgi, right? And that's that one that's going to help us distribute this thing. In her video, she talked a little bit about how this Golgi is going to give us that little extra modification. So it's going to make that protein add sugars, it's going to help fold it. And here's where we use the Golgi. But the cool thing about it is, when this is the ER, right? This is when we make a little fat bubble with some little vesicle. And this is the one that kind of gets pinched off. And when we get to the Golgi, which normally they draw it like this. This vesicle is then gonna, the content of this is gonna bust open like that. And this is gonna end up inside of the Golgi, right? And then it's just gonna keep doing the same trick over and over, where you make a little vesicle, you let it kind of puff up from this side, you cut it, and you connect it to the next branch. And this is connected, sorry, cistern, and then to the next cistern. You literally move this across. And when you're done, well, no, you're not done, you know, you're done with the Golgi, you're gonna again repackage this thing into another little vesicle. And then I want you guys to think of your plasma membrane, right? So this is like, we're getting to the edge of the cell. So this is the cytoplasm, and then this is outside, right? Now, when this is gonna get here, we're gonna again do the same trick. We're gonna bump into it. The things that were inside the little fat bubble are gonna get dumped out, and now they're gonna be outside the cell. So this is kind of one journey that we can take that's gonna get our stuff to the outside. A little bit. Afterwards, we're gonna talk a little bit about a second journey, right? Which there's a little bit of, like we're imagining there's like different options that the cells can do. We're gonna have a little bit of a split. So you're gonna follow me along with this one. Here in the ribosome, it's hanging out in the ER, right? That's what the thing that makes it rough. But we also have ribosomes that just can hang out in the cytosol. Right, so this one is just basically floating around almost in the water. It's not being into this membrane. It's still gonna be the same trick, right? Where we're gonna have our RNA come into that side. This RNA is gonna go work with this one, right? Now we're gonna make our protein. But this protein is not gonna be cytosolic, right? Because this one's stuck inside of that little chamber. This one is still just floating inside of the whole cell. This then makes this protein able to go to different compartments. So we're gonna have like our little chart of the ones that the places this one can go versus the places this one can go. And what you're gonna see is they're gonna work a little bit differently. Because I'm working inside that little bubble or that little vesicle, I can dump this on the outside of the cell. In this case, because I'm working with this free cytosol or the free ribosomes, I can make the protein in the cytosol, and it's basically going to stay. I can imagine it's going to stay on this side of the cell. That's going to be one of the big differences, and then I'll go a little bit about how we actually put them in different compartments. So I'm going to make a little bit of a list here and a list here of where they can go. And normally the trick is that when you put them in a vesicle, it's easy to send them to something else that has a vesicle. You can just use them together. And then the hard stuff is going to be something like this one, where it's like our nucleus actually has a double layer. So, you know how I know we draw one line, and I'm saying this represents like a phospholipid bilayer like that. Right, and then if we were looking at just the very edge of the cell, this is what we find, right? But that nucleus is actually going to be set up like this. So you're going to have one layer on the outside, and then you'll have a second layer on the inside. Because it's doubled, if you try doing this method, it would come in and it would dump your proteins into this space here, right? But the cell has like this other fancy mechanisms, and that's going to be those little purple dots. Later I'll show you how they're super important, because that's where what it'll do is there it has like a little machine that literally grabs onto proteins and shoves them all the way across these two things, right? So it'll go from the cytosol, grab them, and just basically jam it through this whole thing and put them on this. So if you want a protein to go into the nucleus, you can carry it all the way in. If you want a protein to go into that little space between the two layers, then you can use the vesicle tree. Does that make sense? Okay. Yeah. All right, we'll go some more pictures, and then if something happens, we'll come back, and then we'll talk about it a little bit more. So, all right, ER, let's get to the next one. All right, so again, we're really looking at the ER right now, so we're staying on that top half, right? There's my little simple drawing. Here you can see these are basically the little folds. So this is our phospholipid bilayer. It's a single layer, right? Here are ribosomes, and again, the mRNA gets sent out on the cytosol side. It'll go grab onto the ribosome, kind of go through it, but the protein's going to end up inside of here. Right? What's inside of that? Again, now we're going to be in this environment, right? We're going to be inside our little bubble, our little vesicle, and now that'll tell us how we're going to be able to work. The other one you guys need to remember is the rough. This is because of the ribosomes. The smooth, that's the side where you don't have the ribosomes, kind of like this thing. And that's only where we're making lipids. So AKA, we're building more of these little walls. I like the components. All right, oh, I forgot. Yes, they vocab people, they love this one. They call this the lumen. So the inside of our ER, make sure I should probably make this bold. They love to use that word to tell us like, hey, we're inside of the ER versus out here we would say this the cytosol. Inside is called the lumen. So all this little gap here. So we'll find all the proteins. In this class, I think later on I'm going to go over a little bit about how the protein gets made, like as a process. But if you guys take 233, there we go heavy into it. Like you can actually go bring up the diagrams of how the ribosome builds itself and then puts TDN into it. But for us, just to stay on the surface with this, where we're getting the proteins made in the rough side. And here's the drawing I was telling you of what we're going to get to. Right? So this one here on the right, that's the stuff I was drawing over the top, right? So it's like, there's our ribosome is the brown one, the blue horizontal one, that's our mRNA. And then this is the amino acids being glued together to give us this final protein. Right? In that case, on the board, what I walked you guys through was, we go through here, make the protein in the ER, right? It goes through the Golgi, and then we can get it all the way to here, the plasma membrane, right? And it'll be a thing that can dump outside of the cell. Just like that, these would be the other places where it kind of works the same way, but we can get that mechanism to happen, right? Which is because you're in a lipid bubble or a phospholipid bilayer, you can work at things that have one. For the vesicle, it's just a little bubble that you're going to use to dump things outside. The endosome and the lysosome, it's a single layer thing where we're going to build up proteins inside to get rid of everything like on the inside axis like a strong degrader. So all of these is we have to go through the ER to be able to get to these spots. On this side is where it gets a little complicated, right? So this free ribosome, this is when we're talking about basically this pathway, right? Once you've made the protein, when it's done, it's still hanging out. You can see by the color in that same environment. We're sitting in the cytosol. versus we're not sitting in the lumen, right? Because this is floating around, this one could potentially go to different places. From here, you can ignore this one, I don't like it that much. I love to talk about all three of these. And the reason that I like to talk about them is, again, if I did a drawing like this, a vesicle would look just like that, like one single phospholipid body here. If I talk about the nucleus, I already told you guys, right? It's gonna be, it'll look like that. One layer on the outside, you have a second layer on the inside. What do you guys think is gonna happen with the mitochondria and the chloroplasts? Yeah, I can't spell it. But it's gonna be the same thing. Mitochondria has two layers, chloroplast also has two of those phospholipid layers. Right? So again, because if we want to get stuff all the way to the inside in here, you move across two different things. This is where we're gonna have to use this cytoplasmic one, and we're gonna have mechanisms to basically jam the protein all the way through from the outside and get it in, like that. Right? So when you guys think about these are my favorites, which is if I say, hey, how do I get a protein to the mitochondria, the inside? It's the mRNA hits the ribosome makes the protein the cytosol, and then we can take it to the mitochondria, chloroplast, nucleus. That's all the same. On this side, right, it's like, hey, if I want to get this into a vesicle or outside of the plasma membrane, then we know we have to go through the ER. Yeah. All right. All right. Sorry for keep going back to this one. So smooth ER, again, I don't emphasize this that much, but it's just we're making the phospholipids here, we're making lipids. The main thing I like to start with is the protein production. All right, so we're walking ourselves down, right, for this pathway. ER, and we need to get this into the Golgi. So the Golgi, they'll have like these little like flat. Economics back to like flat vesicles, right? So super long, flat, and they'll line them up. Kind of, some of them will be closer to the ER, some of them will kind of be further out. And as you grab a protein, it literally gets popped into that first part. That's going to be near the ER, and then it'll progressively keep doing modifications to it. Normally, it's add sugars, gives it time to fold. If it folds properly, it goes to the next step, where you can get other things like lipid modifications, or you can phosphorate all these events to help fold it. If you mess up, it's also good at throwing stuff back and saying, we do this before we fold this protein. But the main cool things I'm gonna talk about later, not focus too much today, is when you make the fat bubble, there's really cool mechanisms right here that help grab onto the vesicle and they literally push it into itself. That way it can merge both of them. And then normally on these sides here, when they're trying to get rid of the proteins, like this the next thing, it's literally this really cool thing where there's proteins sitting on the plasma membrane that grab onto the protein we care about. Once we build up enough concentration, there's proteins here that'll fold that thing into itself and give you that right. And then there's proteins that will carry this away. Right? So what it's doing is it's doing modifications to the proteins. Like if I went in there and saw this, that's what I would be seeing. Okay, there's sugar being added, blah, 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 blah. But that emergent thing, the big thing coming out of it, it's going to be this. That once they're modified and folded, it has all these little intricate things that come together, and it can send one protein to a specific vesicle, or it can send it to the plasma. And I'll show you guys some of those a little bit later on. All right. So in the quiz, I'm gonna have you guys tell me one of these pathways. I'm not sure which one yet, but you guys might have to follow one up. So I really want you guys to keep feeling comfortable with this story, right? Which is there's our nucleus, right? Here we have we're trying to show like, let's say how the cell's gonna make antibodies. Right? Here we're in the nucleus, you're gonna send out your mRNA, you're gonna hit your ribosomes, right? You're gonna put your antibody in the lumen of the ER. You're gonna make a vesicle, pop it off. It's gonna go mix with the gold. Right? the carbohydrates are gonna be added. That's gonna be the modifications. At the top, it's once it gets added and we know the protein's properly folded, there'll be stuff that catches it and helps build the next vesicle. And that vesicle then gets literally carried into the wall and then it gets dumped out. Yeah, so that's when you guys feel comfortable with. So that was all the production side, right? It's like, hey, let's go from DNA to getting a protein out there to do a task. The other side of this is our cells are pretty cool that they actually can recycle things, right? So now it's time that we don't need these proteins anymore, so we gotta clean them out of the system. And for that, the big player is gonna be our lysosome. So in this case, you basically right now you're following that purple one. Right? And then these little blue things are actually proteins that help clean different things. So some of them can clean lipids, some of them can clean amino acids, some of them can clean sugar. The whole goal of this thing is that whatever you made before to get something to work, like even that antibiotic, if you don't need it anymore, your goal is you need to cut it up and get rid of it. So these enzymes actually will go and you'll have these two things fused. So there's the food that which is absorbed, there's our lysosome. Once it fuses, these enzymes kind of get released into the space. And the other thing that is really cool about these is that they're pH dependent. So before they get, as kind of they're going through this process, they start with a high pH, and as the pH drops, it makes all the enzymes work better. And that's when they start doing the really fancy stuff of breaking stuff down. Like you see, breaking small components. Here, there's our mitochondria. This is going to start getting rid of the fats first, to get rid of the bilayer, and then it starts breaking down everything on the inside. Yeah. So, at which point, if you imagine like this whole story here, right? It's about how we're organizing amino acids to dehydration, right? So you're taking out water and you're gluing the chain. Once you make it to this side, you're doing the opposite. You're bringing in the water, you're breaking the bonds, and now you have individual amino acids that technically look exactly the same as the stuff you were using to put this thing together. Yeah. So that's our way of that same molecule, we can just put it back into the system and use it up again. And some cells is pretty cool because they do that in response to like stress, or like if you start them out and you need to adjust their environment, they'll literally dissolve other things, take out their amino acids and recycle them so they can try building whatever the new things to help them survive. So that's pretty cool. All right, so lysosome digests things, and then these two, another one. People love these two vocabulary words, right? So this is just really tells us where the stuff is coming from. So in the top case, right, this will be the cytosol side, and then this on this side, like the darker orange, we're basically outside. So it's coming from outside the cell. It's phagocytosis. Whenever you eat stuff that you already have on the inside of the cell. So like, you know, your mitochondria inside your cell, that's autophagy. So you're eating your own things. Yeah, so keep this in mind. Vocab, these are some really good ones. All right. So here's a little table with kind of like the things we're going to, right? And you can see the things that I really kind of emphasize from here. It's going to be that membrane is going to be a big issue, right? For the nucleus, it's that double envelope, like two phospholipid bilayers are going to be this thing that's going to affect how we work. The next big player is going to be your ribosomes. Remember here you have two different types. You have your free ribosomes, or you have the ones inside your kind of stuck to the ER. And then, all right, we're getting through the rough. We jump over to the Golgi. This is fine, and we get ourselves to the and the story is going to be that lysosome. Are you guys feeling about this pretty good? Yeah. All right. Should we watch the video again? Or you guys pretty good about it? Like I say, go home on Brightspace. You can download the PowerPoint and you can actually play it. Just look at the video and then think about again, kind of what compartment you're in and where your protein is going. Right. If you have any questions, come back to me and we'll talk about it. Okay. All right. So let's shift into these two guys then. Our mitochondria and our chloroplasts, which are, like I told you, they're going to be a little bit different, so we got to work with them a little bit. So I've seen this people get tripped up with this one. So the mitochondria, this is the one that we use to actually make ATP. And ATP is literally like the gasoline of the cell. Like this is the thing that every enzyme, every molecule loves to burn off in order to steal energy from it. The trick I've seen people get is that mitochondria, you have them in both animals and plants. So this is going to be in both, right? And the opposite to that one is going to be this chloroplast, right? So this one, we only find it in plants and you find it in the algae, but this is the one where we're doing the opposite reaction. We're using sunlight to glue CO2 to make sugar molecules. That's the energy state. And normally what happens is that sugar goes to your mitochondria, and as we cut that high energy sugar molecule, you release a bunch of ATP. So that'll be our little cycle of this thing going through. But mitochondria, animal and plants, chloroplast, only in plants. So when we look at the cell, that's the one I kind of really want you guys to appreciate, right? This is your animal cell. There's your mitochondria. And what we're doing is we actually start breaking the sugars in the cytosol, and it starts getting pumped inward. Once we're in here, and this is the lumen, all the way to the center of the mitochondria, there's where you're really going through the chemical reaction of breaking 6 carbons. Sorry, it starts with 6 carbons here. It'll get to 3 on the inside. And we literally cleave 1 carbon off at a time. And every time we're cleaving up a carbon atom, you actually just are releasing a bunch of energy in the form of like charged electrons. Those are the electrons we're going to use to make the ATP. So that's going to be our little process there. In plants, again, there it is, the fun house, right? Because it needs to be able to grab sugar and create ATP. It doesn't make any ATP from this little part of the chloroplast, which is the one that's, again, turning light into sugar. It doesn't make ATP. aka it doesn't have gas. So it needs basically a mitochondria to be able to burn it off. You burn the sugar, and that's where you get your energy. So when it comes to the story of that protein moving through the ER, through the Golgi to get it to the plasma membrane, it's pretty much the same process, whether you're talking about animals or plants. Right? You can imagine there's a nucleus, we'll make our RNA, it'll travel to the ER. Here we'll make the protein in the lumen, we'll butt it off to the Golgi does all the processing, and then boom, we can send vesicles. So in that case, those are going to be the same. All right. Yeah, so this is a little bit more zoomed in about that mitochondria. And then so again, the big thing I want you guys to emphasize here is remembering that this thing has an inner and outer membrane. So there you can see this is the outer one, and then you have this inner one that has all these little folds into it. Because the cell is doing like, I'm gonna go into the detailed how it makes the sugar, but you guys don't have to write notes on this part, right? So in here, you're breaking up the sugar molecule all the way in the lumen, in the matrix, right? So we're on the inside, there it's breaking the sugar molecules. And what we do is we send the charged electrons towards the plasma membrane. So we build stuff up basically on this wall here. Because we're doing so much work there, the way this thing does it to be more efficient is it folds itself a bunch of times over, aka increasing the surface area, right? So within that same volume, if it was only a circle, we'd have that much surface area to work with, and that would limit how much processing we could do. But by folding into itself, it bumps up surface area, bumps up the amount of enzymes you can have, bumps up the amount of energy you can pull from this thing, right? So that's pretty cool. And that's why it's basically folded, right? It's like we need to bump up the surface areas, give us more things, like be more efficient at giving us the energy. And like I said, a lot of the enzymes that are helping, again, break down the sugar, give us energy, they're going to be found basically on this inner wall here. And they'll be distributed there. Later on, I can't remember in this class if I make you guys go through them, like follow an electron, but 233 for sure, you're going to be like, some of my other classes, they hated me for it, about like, you got to follow the electron through all the different molecules. So that'll be pretty cool. The other one that I like to think about is for example, like the mitochondria, it has its own little bit of DNA, right? So later on, I'm gonna show you guys how they think this is the way that these things evolve, but this thing's holding on to DNA in the center of itself. Because they think it ate a bacteria that needs to burn off sugar to get energy, and that's how our cells adapt to it. And then it really, to me, connects back to this whole story. So one of the things I want you guys to kind of know is like, when you're doing these little vesicles and you're making them, the proteins that are helping build the vesicles, they're corralling molecules into a pocket, right? But there's no control of what's inside that pocket. The stuff that I grabbed on the edge is great. We got them, know what's there, but on the inside, technically, like in here, I'd be like, if I'm trying to hug the whole class, I'd go this way. I give it a feel of these people on the outside, but I wouldn't know who's on the inside. So this is a little bit of a dirty way of making proteins, right? So when you're trying to, let's say, cluster up all the proteins for your antibodies to get them out there, there could be other proteins that are being made at the same time that accidentally sneak into this, right? Let's say a lysosome sneaks in. When you get out here, the lysosome gets left out. Right? It doesn't matter. It'll just dissolve, or it'll take, or it'll break, or it'll try to break things. On the outside of the cell, who cares, right? On this side, if let's say we had that dirty mechanism sending vesicles into our nucleus has DNA. That's the most important information you're going to get. You don't want anything that's a DNA cutter or protein cutter to go into here because it's going to mess with your DNA. And that's what we protect at all costs. So this is where we make the proteins out here, and then we have to invest a little bit of energy in. We literally grab one protein at a time and we jam it into the nucleus. We grab the next one and we jam it into the nucleus. Right? So there's DNA here, and DNA here, and this is where you use that cleaner method of actually grabbing them and bringing them in one at a time instead of just bringing them in huge groups. Yeah. All right. So the DNA is the way that I remember them. So mitochondria has DNA, and also so does your chloroplasts, right? So this is the one that's basically doing the what's called turning light to glue CO2 together to make sugar. And there we have those little pieces of DNA. And then again, that's something where we have DNA on the inside. We're going to be very precise of what we let into that, right? And this is where we're going to control it, where we make the protein in the cytoplasm, and then we literally carry it in. I might put some pictures at the end of this slide today or tomorrow, just so you guys can look at how cool it looks. But I'll make the backgrounds red, and that means you can ignore them. Just if you want to enjoy them, there'll be red stuff. I might go back to my slides and put a few purple ones. If it's purple, that means it's good. Like, hey, focus on this thing. You know? So, yeah, super cool. This is going to be the structure. The green stuff is just those are the proteins that absorb. They're really good at absorbing red and blue. And because they absorb the other two, the one that gets reflected is green, and that's what we see green in the plants. But it just means that they're so good, they suck up all the other colors, right? And as they're doing that, that energy, again, helps the electrons, you move sugar together, and that's how we can make our energy come out of these guys. So again, kind of a little bit near the end now. These structures, they were kind of interested in trying to figure out where they came from and how they evolved. And this is kind of the theory, like, we have like our early form of what eukaryote. We think our first thing was going to be this one, started making our nucleus. And then our cells were probably going through this thing of eating bacteria, right? Like it's not autophagy, it's phagocytosis, right? It's going to eat stuff. When it ate it, that's what they think the theory is, that you see the light orange, that's the plasma membrane to our cell. And then the dark red is the plasma membrane to the anaerobic bacteria, aka the one that burns the sugar. So when it gets made into the mitochondria, then now we have a plasma membrane on the outside, which would be ours, plasma membrane on the inside, which is theirs, and then all the way on the inside, you have their DNA. So it's kind of like, hey, it's its own little thing. Same thing here, here's a divide, but instead of absorbing the mitochondria, we're bringing in that chloroplast, right? Again, light orange, that's our plasma membrane. The darker green, it's their plasma membrane. And that's how we end up again with this double plasma membrane. And inside of that, we have DNA. So the theory is those organelles have their own DNA which tells them what to do. So they can actually make a little bit of RNA. They can make their own proteins to kind of help sustain their systems. This other stuff up here, like your ER or your Golgi and your vesicles, they don't have any DNA. And they're all dependent on the DNA being held inside the nucleus. That'll be the thing that'll make, to replace them, to send signals, all that stuff, has to come from here. These guys, they can almost talk to themselves. So that's pretty cool. All right. So peroxisomes, I normally don't go into them that much because I really love to emphasize that story over there because it goes our central dogma, and we see how all these little things are coming together to give us a really precise mechanism. But for this guy, a peroxisome is like, when you guys think about it, I want you to think of oxygen, right? That's part of the name. So the big thing we're going to be doing here is going to be oxidation. Oxygen is super electronegative, aka it loves to shred things, right? So when you put your H2O2, this is going to give you a little bit more power because normally oxygen with 2 hydrogens, that's water, and it'll be a little electronegative. Now you're popping in a second oxygen and you have 1 hydrogen each. These two are going to be a lot more powerful. And that's really what's going to make kind of shred them. I don't know if you guys, so when I was little, I used to get hurt a lot, like cut myself. And then my mom would buy this thing, hydrogen peroxide, and she'd put it on my cut, and the thing would just bubble up, right? And I was like, oh, what's going on? I didn't know, but now it's like, oh yeah, the molecule is basically breaking up, and whatever's there is going to start breaking. So this bacteria, it's gone, right? So the membrane will dissolve things. So super powerful stuff. Again, this I'll emphasize on it, super low. To be honest, I normally don't like. All right, any questions? No? Good to go? All right. Awesome, guys. See you guys on Monday. And then, again, please come ready for the midterm on Wednesday. That way you guys will have really good questions.